recording in progress.
Hello everybody, I am Claudio Ametrano from Film Museum, and today I want to share with you the result of our project. The aim of the project was to achieve a universal probe set in order to reconstruct the phylogeny of any Filamentous Ascomycetes. Little bit of background on why we choose targeted capture among the whole possible high-throughput sequencing approaches. The most important reason is probably because this method is able to gather DNA sequences from herbarium samples that are characterized by degraded DNA. It can also produce complete coding sequencing if the base set is designed this way. Therefore, the marker retrieved can be useful for other purposes than just build the phylogenetic tree. It can be also cost-effective in comparison to an approach involving whole genome sequencing, and it has both broader application than method based on conserved region, and it's applicable to phylogeny on a deeper time scale than methods based on restriction site or RAD6 that are suitable only for shallow phylogenies. Targeted capture was successfully applied on lichen, and there is a probe set that is designed for lobarioide. The same base set was successfully applied to reconstruct the phylogeny of family Parmeliaceae, and the method was also tested on herbarium specimen which differed in age and conservation technique, and this was carried out on plant tissues. If we really achieve a universal probe set for Filamentous, we could exploit herbaria and culture collection as an affordable source of DNA material for any phylogenetic project. The project is almost complete. We are actually testing on herbarium specimen our base set, but in the beginning we started retrieving a suitable marker for phylogenetic reconstruction. To do so, we both started from annotated genome. We also tried the approach which starts from draft genome assemblies, applying a gene prediction algorithm to delimit orthologous loci, and then only selecting the ones that are in single copy and therefore suitable for phylogenetic. Anyway, there are already established orthologous protein coding gene databases as orthodatabase and the convenient Busco pipeline that is able to pull almost 2,000 semi-universal single copy orthologs from any Ascomycetes fungus. So we started from this genes dataset to select the marker for our probe set. The first question to answer was how much information is really needed. So we build a pilot dataset with almost 200 samples from Pezizomycotina clade, and we reconstructed phylogeny by concatenation, progressively adding markers until the whole amount of genes were used to reconstruct the phylogeny. And then we compare both the topology produced and the average support values of the reconstructed trees. As you can see from the distance matrix and the graph, when you achieve a few hundred markers, both the topology produced and the support values quickly approaches the one produced using the complete gene set. So we can be confident that few hundred markers can be suitable to reconstruct a reliable phylogenetic tree. We were also interested in understanding if there were possible alternative dominant topologies, or if we were in presence of a single locus topology that were very different from anything else in the gene set. To do so, we build a distance matrix, and then we applied an MDS to reduce the number of dimensions, and on this MDS two-dimensional plot, we applied a clustering approach to delimit possible outliers. We did the same with a different approach using a classical outlier test, and we were able to discard more or less the same loci having a single locus topology that were much different than anything else in the data set. It is worth noting that most of these possibly discarded loci are the one from genes having lower substitution rates and possibly not carrying out phylogenetic signal to be used as single locus markers. After this test phylo dataset, we build a dataset much bigger using every Pezizomycotina assembly publicly available on NCBI, and then we only selected for each species the most complete one. We applied the Busco pipeline to pull out the almost 2,000 marker from Ascomycota gene set. We build alignment, and then after building the alignment, we selected them by length, only retaining a little bit more than 1,000 loci. On each of these loci, we applied a k-medoid clustering approach, and the reason for that is that we wanted to minimize the distance between the possible representative sequencing we're going to use for the base set and any target DNA. And the reason for that is as the percentage similarity between the sequence of the baits and the target DNA increases, the number of captured sequence quickly decreases, increasing the amount of missing data. So we had to play with a short blanket, adjusting both the maximum distance that was allowed from any representative sequences and the maximum number of representative sequences that were allowed to build the clusters. We applied this clustering approach, varying the number of medoids allowed and the maximum distance allowed for any sequence in the cluster. And finally, we achieved more or less 200 markers that were able to converge with 1 to 50 representative sequencing. Most of them are slow evolving genes, of course, that were converging way easier, and applying a maximum distance that was lower than 0.3. Then, from this gene set, we excluded 10 genes that were possibly players producing really different really farther away single locus topologies. And we also excluded 3 more markers a posteriori because they were rich in repetitive region, creating problem in the alignment phase. We ended up with almost 200 markers. The original sequences corresponding to the representative sequences were manually created to remove introns, and the sequences were confirmed by well-annotated Aspergillus genome sequences and with sequence from transcriptomic. To test our 200 markers, we built a pipeline that was able, of course, to process target enrichment data using iPiper from Matt Johnson paper, 2019, but also able to accept genome assembly as a starting point, whole genome sequencing data. And probably the most interesting characteristic of our pipeline is that you can also query any taxonomic rank from the NCBI taxonomy to pull out our 200 markers from any sample that has been submitted to NCBI as a genome assembly. This pre-computed database allow you to add sequences to your target enrichment sequencing to build a more complete phylogeny. The rest of the pipeline is a classical phylogenetic tree reconstruction pipeline, producing FASTA file and with an alignment step that is a codon overs for protein coding genes that then applies a segment-based filtering step and an optional block filtering approach. The alignments and then used to both for concatenation and maximum likelihood tree reconstruction, or to use the single locus maximum likelihood tree to build a species tree with the Astral software. After the building of our pipeline in a wrapper script, we were testing our pipeline and the gene set on different data set from literature, starting from a family level phylogeny of Parmeliaceae. We used the same data set as in Pizarro 2019, but instead of using the 2000 loci they used, we used our 200 markers, and then we compare the topology. As you can see from the distance matrix, the maximum likelihood tree retrieved with the concatenation are very similar to the one in the original paper. The same is true for the class level phylogeny we built on Ascomycetes dataset from Aridas et al. 2020. And probably the most interesting result is the one comparing the different topologies produced on the Pezizomycotina phylogeny from Lee et al. Using about 700 genomes. We tested our gene marker also on this data set, and as you can see from this MDS, from this two-dimensional MDS, the original phylogenetic tree reconstructed in that paper are basically clustering by reconstruction method. This cluster is the astral method, topologies, and instead this other cluster represents the phylogenies produced by concatenation. Our three are different in topology, but the distance is again pretty small. If we go into details comparing the logical mismatches, the family level phylogeny from Parmeliaceae present just minor mismatches. Only the Relicina intertaxa placement is really different than between the original tree and our tree. What is important to note is that instead the tree produced by coalescent using astral present a high level of conflict in the nodes, and the level of conflict is here highlighted by these pie charts. The red part is represents the nodes that are presenting different conflicting topologies. The green part is instead when there are conflicting topologies, but just two of them, alternative topologies. And when the pie is blue, we have concordant topologies. So the level of conflict is particularly high, and this is because most of the genes are producing a single locus topology that are different from each other. The same constitution are valid for the class level phylogeny, and there are few topological mismatches, and most of them are from taxa that are known to be difficult to place within Dottyomycetes. For example, Eremomycia is a known row taxa in Dottyomycetes, and also Coniosporium apollinis is still Incertacetes in the class, and this is also highlighted by the comparison between the original tree from Aridas et al. And our tree built using our 200 markers and our wrapper script. The same consideration we did for the level of conflict on the nodes of phylogeny are valid for the class level phylogeny, with a high level of conflict in that is very widespread along the nodes of the phylogeny built by coalescent-based approach. To conclude, some take-home messages and possible future development for this pipeline and this approach. The marker set proved to be effective. The phylogenies that we reconstruct were mostly concordant to those produced using much more data. And as expected, the concatenation produced the best result. And this is because on one side, the single locus tree produced very different topologies due to the fact that we were also mostly using slow genes that were like, easy to represent with less representative sequences. And another reason is because we are also using a number of loci that is kind of in between a classical multi-locus approach and a real genomic approach. We could try to subset our 200 genes to decrease the amount of conflict in the astral tree, but we have like kind of a small room for maneuver because we are already starting from kind of a low amount of lossy, only 200. The efficiency of the target enrichment approach must be, of course, tested on a real case study. We're gonna test our probe set on lichen herbarium specimens, but we are also working on to develop a mode of our pipeline that works as a multi-locus barcode approach, and this will be possible because the genomic resources that are becoming available are quickly increasing, and therefore the database used to apply our markers as a multi-local code is becoming stronger and more reliable. The universal base set, the wrapper script, and the pre-computed database will be available soon. The base set will be designed in partnership with Arbor Bioscience, and will be available as a pre-designed panel on their website. The wrapper script and the pre-computed database will be instead soon available on my GitHub under the name Unfate. That is Universal Filamentous Ascomycetes Target Enrichment Approach. I want to thank Kathleen and Jacob for the help they provided in developing this pipeline, and of course our funding source. Thanks for listening.